Well, thank you, Sandy, and thanks for everybody who's joining us celebrate the 5 years of the Biohub. Next slide, please. So I'm actually going to start where Angela left off. What she's just showed you is that really the defining signature for all the cell types in our body are the sets of genes that these cells express. But of course, it's not the genes themselves, which are the molecular building blocks of the cells, but rather these are the proteins that the genes encode for. And so next slide. What genomics, in a way, really provides us with are part lists of all the components that make up the cell. And now the opportunity for cell biology is really to try to figure out the rules of how all of these components come together to build the human cell. And of course, this is a tremendously difficult question because there is 20,000 different proteins, more than 1 billion molecules of protein. There is still so much we don't know about how the cell is built. Next slide, please. So to help fill that gap, the strategy that my lab is taking is to deconstruct the proteome or the cell one protein at a time. And to do that, we use CRISPR to rewrite the genome of the cells and turn these proteins fluorescent by using fusions with tools like GFP. And that allows us to measure two things for each protein. Number one, where proteins are localized within the cell. And of course, we can do that using live cell fluorescence microscopy. But we can also measure the interaction, the molecular interactions between these proteins. To do that, we use the fluorescent portion as a purification tag. We can extract a protein of interest from the cell. And then we can use a technique called mass spec or interaction proteomics to ask what are the other proteins that are coming along for the ride? Again, defining these molecular connections. So you can imagine that conceptually, if for each protein in the cell, we could measure its localization and its interaction partners across the proteome, we could really map fully the wiring diagram of the cell. Well, of course, that's going to keep us busy for some time, but next slide, please. Through really a heroic effort by many people in my lab over the years, especially under the leadership of two great RAs, Nathan and Preeti, we've done this experiment now over 1,300 times, and we generated OpenCell, which is the biggest paired live imaging and interaction proteomics dataset currently available for the human proteome. And I should mention that all of the mass spec was done through a fantastic partnership with Matthias Mann at the Max Planck in Munich with two of his students, Andreas and Andre. And all of the data I'm going to show you today is coming from the HEK293T cell line. So next slide. This is what our data looks like. On the left side, you have live cell 3D confocal microscopy images. As you can see, there are many cells in this image. Their nuclei are labeled in blue and the protein of interest is in grayscale. And on the right-hand side, you have a graph that represents all the connection that our protein of interest is making molecularly with other proteins in the cell. Now, next slide. To really make sure that our data set would be fully accessible, but also reusable for the entire community, we went out and built a fully interactive website. And this brilliant work by Keith, who is a software engineer on the team. And just to illustrate our interface, if we could please play the video. You can search for your favorite gene. You can explore the localization of the corresponding protein in full 3D. And then on the right-hand side, you can click your way through this interaction graph, really jumping from one interacting protein to the next and starting to really navigate the inner wiring of the human cell. So I know we went kind of fast, but again, you can go to our website, explore all of our data. You can also download all of the raw data. You can actually also download all of the source code. And really what we try to provide is a fully open source tool that we hope is going to be useful for a variety of applications across the community. Now, of course, now that we have this critical mass of data, what is really fun is to analyze it. And we were especially interested, next slide, please, in sort of like holistic analysis, looking at the structure of the whole data sets in a way that would tell us something about the global features that organize the human cell. Here's an analysis that I particularly like that was derived by Kibom, who is a data scientist on the team. What you're looking at here is the full hierarchical structure of our interaction data. And if you look at the tree on the left-hand side, you can see that there's a lot of organization in this data set, but especially there are two groups of proteins that really stand out. In the middle here in blue are proteins that happen to be related to membranes. And to the right of it in green are a bunch of proteins that bind RNA. And what we went out to show is that we think What really segregates these two groups of proteins from the rest of the cell are specific biophysical properties, namely hydrophobicity for membrane proteins, which makes total sense, but more interestingly, intrinsic disorder for RNA binding proteins. And in a nutshell, what we think this analysis is showing us is how evolution is probably co-opting different biophysical features to globally shape cellular architecture. So I apologize that I don't have time to do justice to this analysis, but rather I wanted to spend more time, next slide please, telling you about what we learned about the spatial organization of the human cell through our data set. Now, as you know, classically, the architecture of the cell has been described across a dozen or so different compartments. These are things like the nucleus, the plasma membrane, and so on. Now, this framework is really interesting, but the semantics that it offers are rather limited. Especially, it is kind of hard to describe nuances of localization of proteins between different compartments. It's also very hard to truly be quantitative when we compare two proteins together. And so, next slide. What we decided to do is to turn to machine learning and deep learning in particular, which, as you know, is completely transforming our approach to image analysis. And through a really fantastic collaboration with my colleague Loïc Royer at the Biohub and his postdoc Hero, we went out and built a new kind of deep learning algorithm, which we call CytoSelf, which importantly is completely self-supervised. What that means is we're not using any kind of manual annotations to train the model, but rather we're only providing the images and letting the model figure out what is unique to each protein and how to best represent it. So if you want to know more about all the bells and whistles that we had to develop to get this to work, you can go and see our preprint CytoSelf on bioRxiv. But essentially now what we have is this tool to transform images into quantitative encodings. And now from these encodings, we can apply all the suite of data science to analyze this data set in a very similar way that our colleagues are analyzing single cell RNA-seq data. Next slide. So what I'm showing you here is a UMAP. This is a 2D representation of our data set, which is very similar to the maps that Angela was just showing you. But here, each dot is one protein in a cell, or rather the spatial distribution of one protein in a cell. You can see that there's a lot of structure to this map. And when we layer the annotation for classical compartments, well, these define very delineated areas on the map. The nucleus is on one side, the ER is on another, the cytoplasm is yet on another. And that's really great because that means that our machine learning encoding is really deriving a sort of like biologically meaningful representation of these images. But as you can also appreciate, there's also a lot of fine-grained structure, a lot of small clusters in this map. And that led us to ask how much we could exploit the structure of this data set to extract specific biology. And in other words, how much of the known organization of the human cell could we recapitulate just by doing image analysis? Next slide. And to our surprise, the answer was actually quite a bit. So what I'm showing you here is the result of an unsupervised clustering of our data. What we're doing here is we're grouping proteins on the basis of similarities between their spatial distributions. And we can do that at different degrees of resolution on the x-axis. Increasing resolution is going to define smaller and smaller clusters that are going to group together proteins that share finer and finer details. And on the y-axis, we compare these clusters to known sets of annotations. And I'm just going to say that all these values are actually pretty high. So what you're looking at here is that at low resolution, our clusters recapitulate really well known cellular compartments. And that makes complete sense, on the basis of what I just showed you. But what's really interesting is when we look at higher resolution, right, we can recapitulate with almost the same performance biochemical pathways all the way to protein complexes, molecular level information. And really what that's telling us is that there's a ton of specific functional relationships that we can derive and interpret on the basis of imaging data alone. So let's look a little bit more at this example of protein complexes. Next slide, please. So what I'm showing you here are different subunits of giant molecular assemblies, things like the ribosome, the nucleopore, and so on. And you can see that these subunits from the same big complexes fall right next to each other in image space. Next slide. And when we look more globally at all the different proteins that happen to interact within our data set, And we plot on the y-axis the similarity of the spatial distribution against the stoichiometry, the stability, the strength of their interaction on the x-axis. What you can see is that most of these proteins don't really share a high degree of spatial distribution similarity. In other words, the distribution of each protein is pretty unique to itself, with the exception of this group of proteins at the top right-hand corner, which is indicated by a dotted line, which are proteins that have very high similarity of spatial distribution. And these correspond to proteins in the cell that form very stable interactions. So for the stable interactors, the localization features become indistinguishable. Next slide. Now, what's interesting is that we can flip the argument over and ask, well, can we actually predict molecular interactions just on the basis of comparing images? And this graph shows you that indeed it is possible. So on the x-axis, as we look at any pairs of two proteins, at a certain level of similarity of spatial distribution, we can actually predict with high probability that these two proteins will interact. Next slide. And just to illustrate kind of an application of this idea, we took a protein that was completely uncharacterized. It's called FAM241A, fairly barbaric name, which again, represents the fact that nothing was known about this protein. We tagged it, we measured its spatial distribution, and we fed that to our machine learning algorithm, which came up with a very specific prediction, which is that this protein would be part of a complex called OST, which is the glycosylase at the ER membrane. And very satisfyingly, when we did the control experiment, which is doing classical interaction proteomics, this is shown at the bottom now, we came up with exactly the same answer. So it is indeed possible to predict molecular interactions with imaging data alone. Next slide. And so if you think about everything that I've told you, really the picture that emerges is that the spatial distribution of each protein in the cell is very unique and really distinguishes proteins from one another. And it's only when proteins are part of the same biological functions, pathway or complex, that their spatial distribution starts to be very similar. And the manifestation of that is that when you start now looking at these fine-grained clusters in our map, which I pointed to before, these clusters really beautifully correspond to specific biological functions. And what's even more interesting is that this seems to be true all the way to soluble proteins. And a very interesting example is this cluster on the very right-hand side, cluster 17, which corresponds to proteins which, under the conditions that we did our experiments, were expected to be homogeneously distributed in the cytoplasm. Well, it seems that rather, all of these proteins have a very defining features in their localization, which the algorithm is able to pick up that distinguishes them from other cytoplasmic proteins. When you think about that, I think what that says is that there's probably a degree of micro-compartmentalization, sub-compartmentalization of the human cell that is much higher than maybe we have anticipated. Next slide. So I'm going to leave you on these thoughts. I've shown you how OpenCell combines engineering innovation for high-throughput cell biology, which led to the development of a large library of CRISPR-Tag cell line. We turned that into a fully open source and web-accessible data set, both of images and interactions. And I've shown you how we're using this data now to develop new computational strategies, including the development of new machine learning algorithms. One of the key take-home messages is that the spatial distribution of each protein seems to be remarkably predictive of their function. And I think that for the few of us that are interested in using microscopy as a phenotypic screening tool to learn about the cell, that means there's really a potential goldmine of information that we can extract from this kind of data set. Next slide. If you want to know more about anything that I've told you, please go and check out our website at opencell.czbiohub.org. You can also read our preprint in some bioarchive. I hope you'll see that in a peer review journal very soon. And last slide, please. I want to finish by giving a huge shout out to all of my team, but also pretty much every group and platforms, the whole Biohub. This was a huge multidisciplinary effort. And frankly, I don't think I could have done this anywhere else. I want to finish with a shout out to my collaborators at UCSF, especially Bohong, and in Germany, Matthias and his team for fantastic partnerships. And thank you for your attention.
Manu, thank you very much for a fabulous talk. With the machine learning based approach for for looking for similarities in images and the UMAP clustering of images. Have you begun to explore ways of generating human interpretable measures of what the basis for similarity is? So part of it is just where things are co-localizing in the cell, but it seems as though there may be other patterns occurring. And do you have a way of teasing out what those patterns are?
Right. This is specifically something that Loic and I are interested in exploring further. We touch a little bit on that in our cytoself preprint, trying to interpret what are the features that the algorithm is really using to distinguish proteins with respect to one another. I think it's the early days, but again, this is a really active field of research, kind of like, opening or interpreting the black box of machine learning. And we think that, there are some approaches that we can take to try to make sense of the black box. But again, very early days, but we're definitely looking at that.
That's great, thank you. So then there are a number of sort of basic questions here. Could you comment a little bit more on how the spatial distribution similarity is calculated?
Right, this is calculated again by looking at the vectors that are provided by the machine learning algorithms and really essentially doing math and calculating correlations between these very high dimensional vectors. I would encourage people who are interested to know more to check our preprints for kind of a full technical descriptions of how we do this kind of analysis.
Great. And then a couple of questions regarding, are you planning to tag more proteins? Are you planning to do additional cell lines? And what was the rationale for choosing HEK293 T cells to begin with?
Right, so the rationale was simple. We had to start with something that was simple enough so that the cell culture itself wouldn't become, a huge bottleneck. But of course, what we're trying to do here is build a set of tools that are really generally applicable and that would be plug and play enough that then we can move to, the next relevant biological system to really start asking specific biological questions, for example, in the context of diseases. And I think that a lot of our effort right now, for example, is looking at stem cells and engineering stem cells and differentiating them to look at specific biology. Again, still the early days, but this is definitely an adventure that we hope to continue.
That's great. Manu, thank you very much again for a terrific talk.
Thank you, everybody.
And Andy.